with the muscular system. This is chapter 12. And this is probably one of my favorite systems, muscle. This is actually an area of my research interest. So muscle morphology, fiber typing, that type of thing is where I typically do research with human cadavers, the histology of muscle, really my lane in terms of research interests and so on. So it's a really exciting topic for me. So I'm going to try to not talk, go on to the tangents that I typically do here. So we're getting into the muscle. We're starting with the physiology, packet 1. And here we're really going to introduce the general anatomy of muscle and then talk about 2 other phenomena. Muscle contraction. So how muscles physically contract, that is called the excitation, contraction, coupling, how we generate the energy for that muscle contraction, how calcium comes into play. And then we're also going to speak a little bit about force generation. So that'll come later on. We probably won't get to this for this first exam, but that'll be included in this first packet. So how muscles contract, the shortening of the sarcomere, the structure of the sarcomere, really the anatomy and introduction of the histological structures there. And then we'll get into the force generation, which is more of the physiological aspects of muscle contraction. All right. So muscles are very important, as you would imagine, right? We've got 3 different types of muscle in the body. We've got cardiac muscle, smooth muscle, skeletal muscle, and we're starting out here looking specifically at skeletal muscle. So we should already have an understanding at this point that skeletal muscle is the part of the muscular system that we can control. We can voluntarily decide to move our hands and eyes and fingers and toes. And this is the type of muscle that we're going to delve into. Later on, we'll speak about smooth muscle and cardiac muscle and their mechanisms of action. So skeletal muscle is pretty much how we walk, talk, the majority of physical functioning. And so muscles are generally suspended across 2 or more bones by tendons. So we typically have a muscle being stretched and suspended in all of the locations that we talk about muscles. So we've looked at muscles, for example, the biceps, that is suspended across the elbow joint, right? And it is held in place, it's attaching to 2 bones. It has a point of origin where it's attaching at the proximal part, and then it has a point of insertion where it's attaching at the distal point. So that is to help the muscle be suspended and kind of be slightly stretched, right? That's how we want muscles to be in their resting state. And they're connected to the bones physically by a structure that's called a tendon. And so the tendon is the non-contractile part of the muscle. And if you look at this image here, we've got the muscle belly itself. I usually refer to this part as the belly or the body of the muscle, whereas the tendinous portion, this white fibrous appearing portion here, is what is non-contractile. So that doesn't move. Only the contractile bit, the muscle belly, moves, but the tendon is what helps to drag that insertion and help to move those bones. That's how I can move my hands, move my feet by the shortening of the muscle and the tendon dragging that bone in the corresponding action. Okay. Looking more closely at the muscle belly or the muscle body itself, there are several different compartments here. We have the epimysium, which is the connective tissue sheath that is overwrapping the muscle. This helps to separate one muscle group from another. This is how I identify my biceps as being different from my triceps, as being different from the deltoid, because of that epimysium that selectively encircles one muscle group from another. The epimysium is also important in helping muscles to slide over each other. So having that sheath that kind of loosens the friction and help the muscles move over each other is really important in that shortening of the muscle as well. Now, within the epimysium, we have the muscle fibers, and muscle fibers are then individually wrapped with the perimysium. Okay, so within an epimysium, we have the entire muscle belly. Within the muscle belly, underneath the epimysium, we have individual muscle fibers, and surrounding each muscle fascicle, excuse me. So again, within the belly, we have fascicles. Within the fascicles, we have fibers. So the fascicles are divided by the perimysium. So it's dividing the muscle into several different fascicles. We have about 100 to 1,000 of those within a single muscle body or muscle belly. And then within the muscle fascicle, we have a muscle fiber. So the muscle fiber is synonymous with the functional unit of muscle. It is synonymous with a muscle cell. A muscle fiber is the same thing as a muscle cell. And then surrounding each muscle fiber is the endomysium. Okay, so just to kind of recap that and reiterate that, we have a single muscle fiber or cell surrounded by an endomysium. Several muscle fibers make a muscle fascicle. A muscle fascicle is surrounded by the perimysium, and then several muscle fascicles make a muscle body. A muscle body is surrounded by the epimysium. Important components of a muscle fiber, that is the cellular structure of muscle, is that they are multinucleated. We'll see for a smooth muscle and cardiac muscle, they are uninucleated, so they only have one nucleus, whereas skeletal muscle, and we'll see with good reason, has several different nuclei to provide for lots of energy and lots of demand. There's also the sarcolemma, so some unique terminology to muscle here. The sarcolemma is the unique name that we term the plasma membrane. So whereas in other cells, we call this surrounding structure the plasma membrane, here in muscle, we call it the sarcolemma. Similarly, the cytoplasm in other cells is referred to as the sarcoplasm in muscle, right? So similar sets of structures, just some unique terminology to muscle cells. All right, so just kind of reiterating this schematic here of what the muscle looks like. We've got the tendon, which is attaching to the bone. The tendon is non-contractile. We've got the actual contractile portion, which is the muscle belly or body here. Within the muscle belly or body, we've got muscle fascicles. Within the muscle fascicle, we've got muscle fibers or muscle cells. And then within the muscle cells, we have these myofibrils. I'll talk more about the myofibrils. This is kind of where the magic happens at the level of this microscopic or histological level. This is where the machinery for muscle contraction exists. Also, something to point out is the several different nuclei. So skeletal muscle specifically is multinucleated, it has many different nuclei, and we'll see some other types of muscles that do not have more than one nucleus. Okay, getting into, again, more of the histological appreciation of muscle here. And if we look inside a single muscle fiber or a single muscle cell, we're going to see many myofibrils. Myofibrils are the contractile units of muscle, right? So they are these elongated fiber-like structures that are running up and down the length of the muscle fiber. And these have repeating units on them called the sarcomere. So if you can see this one single myofibril that's been pulled out here, you'll see this repeating sort of interdigitated unit along the length of the myofibril. This junction here, this one of these little, from this blue line to this blue line, one of those segments is called a sarcomere. And that's going to be really important in understanding how muscle contracts. Other things found within the muscle cell are the sarcoplasmic reticulum, which is, again, unique terminology to muscle. In other cells, we call this the endoplasmic reticulum. In muscle, we call it the sarcoplasmic reticulum. This is the smooth ER, or the smooth endoplasmic reticulum. This is really important because this is where calcium is stored. Calcium is vital for muscle contraction. So we cannot have contraction without calcium, and we'll see that in a minute. But the storage area for that calcium is the sarcoplasmic reticulum. There's also many mitochondria. So energy is a really important demand for skeletal muscle, because our skeletal muscles are so quick in their response, right? They're voluntary muscle. They need to be able to move very quickly. So they need to have a lot of energy to be able to do that. They also are important in force generation, so lifting heavy objects, providing lots of tension for exercise, for activity, for movement. And so having lots of mitochondria is really important for muscles to be able to do that. The other important structures here, the transverse T-tubules. So the transverse tubules, sometimes referred to as T-tubules, are these invaginations that are shown here in green that are running along the surface of the muscle. And although we're seeing them here on the outside surface, they're deep invagination. So they're going to go right down. We can see in this kind of this zoomed in picture here that the T-tubules run into the depths of this muscle fiber. So this is really going to be important in terms of spreading the electrical activity that generates muscle contraction deep throughout the muscle very quickly. These deep invaginations that are called these T-tubules or transverse tubules. The lateral sacs, also called terminal cisternae, are how we can communicate that calcium from the sarcoplasmic reticulum into these spaces where they can be used for muscle contraction. So we'll talk about this region here called a triad. And the triad is where we have two terminal cisternae or two lateral sacs coming into contact with the T-tubule. These three structures together are called a triad. And a triad is going to facilitate that muscle contraction as well, especially as it relates to the storage and the communication of that calcium, so that calcium can be involved in the muscle contraction. And that will make more sense as we talk about the machinery that allows for that. So important things just to reiterate here, the T-tubules, these invaginations, coming into contact with the lateral sacs, which have calcium in them. That structure is called a triad. We've also got lots of mitochondria for the energy, the ATP production to sustain muscle contraction. And then we've got the smooth endoplasmic reticulum or the sarcoplasmic reticulum, which is where we store that calcium when it is not being used. Okay, getting more into this myofibril structure. So we talked about it just a little. This is what gives skeletal muscle its striated appearance. So this is why smooth muscle is called smooth muscle. It does not have a sarcomere. It does not have myofibrils. And so the sarcomere gives this striped or striated appearance to muscle. This is what makes even if you happen to eat chicken or meat or any other type of muscular muscular meat, you'll see that kind of stripped, striated, elongated sort of tendinous appearance, and that is being created by the myofibrils. It's going to be arranged, kind of stacked into the muscle fiber. And if we look closely at the myofibril itself, it has these arrangements of thick and thin filaments. The thick filaments are in the middle here, these orange structures. The thin filaments are at the sides of the sarcomere, these blue structures. And these are going to be really important as far as muscle contraction. These are going to be the actual structures that help to shorten the sarcomere, which will shorten the myofibril, which will shorten the muscle fiber, shorten the muscle fascicle, and eventually shorten the entire muscle, which is bringing about a contraction. Okay, so the level of the sarcomere is where the magic happens, right? The action of the actin and myosin, these thin and thick filaments, is what brings about muscle contraction. Okay, let's look more closely at that sarcomere here. So this is a myofibril, again, not to be confused with the muscle fiber. Many of these myofibrils are stuffed inside a single muscle fiber. So if we took one of those and looked more closely at it, we would see this region that's called the sarcomere. So the sarcomere by definition is Z-line to Z-line, okay? And I'll talk about what these lines and bands are in just a minute, but this central region here is called a sarcomere, and we have many sarcomeres along a single myofibril. So it's really important to start to kind of organize these structures in your head in terms of thinking about a muscle, thinking about a fascicle, thinking about a fiber or a cell, thinking about the many myofibrils that are inside that cell, and then think about the many sarcomeres that are lining that myofibril, okay? Now, if we think about these different bands and zones, these are areas of demarcation of these protein filaments. So this actin and myosin that I just described, the blue lines being actin, the orange lines in the center being myosin, these are filamentous proteins. And these proteins are going to be interacting with each other, but we describe them based upon their histological appearance as these bands and zones. And just to kind of give the backstory of how we initially came up with this description, we had some early physiologists who were first looking at muscle under the microscope. And all that they could identify were just some different staining regions. They saw some light regions like this at the bottom here. They saw some kind of gray in between regions. They saw some dark regions. And so at the outset of kind of investigating what makes up muscle, they just label these regions as bands and zones. So we've got the Z lines, which are the darkest staining regions of the sarcomere. These are also the boundaries or the borders of the sarcomere. So the Z line is here and here. And these are made of all of the connection of our thin filaments. So the blue structures here I mentioned are the thin filaments, which are myosin, which are actin, actin, thin filaments. And so the line that connects these thin filaments is called the Z line. And because we can see that it has this kind of crisscross, jagged appearance, that is where we get that term Z-line from. Another region that they described was the A band. And the A band is this entire span, this entire region here. Now, the A band is made up of an area of overlap. So we're seeing an overlap between the thin filaments, which are actin, this blue structure here, and the thick filaments, which are myosin, the orange structures here. And as you can appreciate, there's an area where those two structures overlap. That region from this end all the way over to this end, that is called the A band. Okay, that's kind of this medium gray staining region histologically. The other region that they appreciated was the H zone. And the H zone is a region in the center of the sarcomere, so it's kind of around here. It's a lighter staining region than the A band, but it's not as light as the I band that I'm going to talk about in a minute. So again, it's kind of an intermediate region, and this is where we have the thick filaments, but there is no overlap between the thick and thin filaments here. So in terms of the schematic, it's the central region here, right? From this end where there's no overlap, all the way to this end where there's no overlap, there's only the presence of these thick filaments, which are along the center here, and that is forming the H zone. Okay, so myosin or thick filaments forming the H zone. And then right along the very center of the sarcomere, the line here is called the M line. This is the line that anchors or joins each of our thick filaments together. So all these thick filaments that are coming across here are anchored together via the M line, and that is this line here histologically. Okay, let's move on to the I band. So the I band is the lightest or palest staining region of the sarcomere. This is only thin filaments or actin. And because actin are smaller proteins, so myosin is a thicker protein, so those thick filaments are called thick filaments for that reason. Actin is a thinner protein, so the thin filaments are called thin filaments for that reason. And because there's only the presence of actin, no overlap, forming the I band, this becomes the lightest or palest staining region of the entire sarcomere. Now the Z lines, as I mentioned before, which are the borders of the sarcomere, so from Z line to Z line, the Z lines are the regions that link those thin filaments together, forming this jagged appearance, which we refer to as the Z line. And then lastly, the functional region or the definition of a sarcomere is from one Z line, the confines of one Z line to another Z line. That region is one sarcomere. And we can see that there are many sarcomeres that are spanning the length of this myofibril. And then each of them is kind of separated by these Z lines. Okay, so just to reiterate those staining regions, again, we've got the Z lines, which are the borders of the sarcomere. These are the anchoring points of the thin filaments or the actin filaments. We've got the I band, which are the regions around the Z line. These are the regions formed only by thin filaments or actin. These are the lightest regions of the sarcomere, right, on either side. So there's no overlap just yet between thin and thick filament. There's only thin filament. We've then got the A band. The A band is the region of overlap. So where the thin filament slightly comes into contact and overlaps with the thick filament, that is where we begin to have the A band. And as you would imagine, we've got two proteins here now. So this is going to have a darker staining pattern in terms of this histological view down here. Within the middle of the A band, we have the H zone. This is the region of just thick filaments. So it's going to be darker than the I band because myosin is thicker than actin, but there's no overlap here. So there's no overlap between thin and thick filament, but it is just the center of these thick filaments right along the middle here. And then the very thinnest part, the middle, absolute middle of the sarcomere, is formed by the M line, which are the anchoring or connecting points of the thick filaments or the myosin filaments along the center of the sarcomere. Okay. Looking at a cross-section of this myofibril, we can get a better appreciation of the difference in the thickness of these two filamentous proteins. So the thin filaments are blue here, very, very thin proteins, hence staining much lighter in terms of this histological view, whereas the thick filaments are much thicker proteins. These are forming the center of the sarcomere and are going to be much thicker in terms of their staining arrangement here, but also along the center of the sarcomere here. Okay. All right, I'm going to pause there and see what questions we have, and then we're going to get into some of the details of these two filamentous proteins. All right. So we've talked a lot about these two proteins, actin and myosin. We're going to get more into the actual structure of these proteins. What are these proteins actually made of? Why is the thin one thin? Why is the thick one thick? How do they look? How do they interact with each other? And how do they eventually generate muscle contraction? So the thin filament formed by actin is the contractile protein. This is the protein that is going to be dragging myosin, okay? And so just to come back here and kind of explain the big picture of this, and then we'll go on and fill in the details. Actin is this blue line here. Myosin is the orange line here. And the way that muscle contracts is by actin reaching up, grabbing onto myosin, and then dragging myosin towards the center of the sarcomere, okay? And so actin is going to be coming into contact with myosin, and then they're both going to be moving towards the center of the sarcomere. Myosin is going to move that way, actin is going to move that way. And so as we'll see, that will generate the Z lines coming closer and closer and closer together. As those Z lines come closer together, the myofibril starts to shorten, and then the muscle fiber shortens, et cetera, until we see the entire contraction of the muscle itself.